Chapter 9-1, The Nature of Blood. So blood typing was first discovered by a man named Carl Landsteiner. He's actually one who is credited with classifying our ABO blood system. Um, Forensic scientists are going to focus on blood factors that are genetically controlled, which means that they're linked or can be linked to an individual. Um, But as the advent of DNA technology appeared, this kind of altered the approach of how we use and what we use blood evidence for. But it also changed how and what type of evidence we collected from a crime scene in order to look for any sort of individual evidence that we can link to an individual, such as blood or now DNA. So let's talk about the nature of blood. So first off, blood is a complex mixture of cells, enzymes, proteins, and inorganic substances. Um, it's the components of the blood that are pertinent to the identification. In other words, these are what the forensic scientists are looking for in order to help identify whose it is. First off, it's composed of plasma, which is our fluid portion of the blood. It's mostly water, but it accounts for 55% of the content of blood. Next are what we call the erythrocytes or the leukocytes, um, which are part of the solid portions of the blood. Um, they include the red blood cells or RBCs and the white blood cells or WBCs, and they account for 45% of the blood. And then the last small, uh, basically, what it's floating in is what we refer to as serum, which is the pale yellowish liquid that when you scrape yourself and that starts to ooze first before it starts to bleed. That's the serum. But on each of the blood cells are what we refer to as antigens. These are usually proteins. Like I said, they're located on the blood cells, specifically the red blood cells, and they are what are responsible for our blood typing. We have actually more than 15 um, blood antigen systems, but the most important ones that we look at are the ABO and then the RH systems. So if somebody is type A blood, that means they have the A antigen on their RBCs. If they're B, they have the B antigens. If they're AB, they have both antigens. And if they're O, it means they have neither A or B antigens. And the O blood type is the recessive. Um, so we have talked before that in biology, where you guys learned where our blood cells or our blood type is a co-dominant trait. So this is why somebody who has both A and B antigens or A and B allele will show both the A and B antigens. The RH factor is determined by a third antigen or another antigen, and we refer to this as the D antigen. And this is either present or it's not present. And so this is where we get our positive, negative from. So if somebody has the D antigen on their blood cells, they're referred to as RH positive. If they don't, then they're RH negative. But for every antigen we have an antibody. Okay, so the serum is the important portion here on determining what our blood type is. And so these antibodies are proteins that float around within the serum. And for every antigen, there's a specific antibody. So if there's anti-A is going to be specific to the A antigen, which in other words means that if there is an anti-A antibody, okay, or antibody A, and it comes in contact with an A antigen, it will link together and start to clot. And this is known as agglutination. And so, in this is the reason why if somebody is um, um, a let's say a B blood type, that means that they're going to have the antibody A. So they're going to have the opposite of what they are in their serum, and that means that if they're given the A um, blood type, then those anti-A's that are floating around in their serum are going to um, attack them basically and clump together and cause agglutination. This is the reason why being given the wrong blood type can be fatal, because the body is programmed to fight off um, the opposite blood type. If somebody is AB blood, that means they don't have any antigens, or excuse me, they don't have any antibodies, and that means that they can receive all types of blood. So they're considered the universal receivers. Opposite to that is the O blood type, who has no antigens, which means they have both antibodies and can only receive O. Um, but also to that, because they have no antigens, that means that they can be given to anybody because they're not going to react with anybody's antibodies. Okay. Um, so, serology is what this whole chapter is on, and it's basically a broad scope of laboratory tests that use antigens and serum antibodies reactions in order to determine different information about an individual. So the biggest one is blood typing. ABO types are established using this anti-A, anti-B sera when they are um, combined with a unknown. If they are that blood type, then it will clot. So if you look here over on the side, if somebody is um, A blood type, and that A blood type is put in with the anti-A sera, then it will cause it to clump. Okay. If it clumps, okay, this is what we're talking about with the clumping. If it clumps over on the right-hand side, that indicates that that's what their blood type is. If it doesn't, then that indicates that it is negative for that antigen. And so if we're looking over on the right-hand side, the circles that are closest to the letters are indicating clumping. So this would be A blood type, meaning that the B does not clump, which means that they are A blood type. This third one here, the blue is the RH factor. So that would be that third or fourth, excuse me, blood factor that they look at, that D factor. So again, if somebody is B, then the B clumps and not the A. I totally said that backwards. I apologize. The blues are the A's, the yellows are the B's, and the whites are the RH. Um, so again, if it is A, then it clumps with A. If the person's B, then it clumps with B. If it's AB, it clumps with both. And if it's O, it clumps with neither. So with the immunoassay technique that I touched on in the last one that I said is a screening test for drugs, basically, assay is any sort of analytical technique that's used in a lab. So immunoassay is looking at the antibodies within the blood. Okay, so we have what's known as that immunoassay technique that's used as a screening test for tools. And so basically, we have a couple of, we actually have a large number of them, but the two that we're going to focus on is what's called EMIT, so enzyme multiple immunoassay technique. This is quick, it's highly sensitive for detecting drugs in urine. And then the radioimmunoassay, which uses drugs labeled with a radioactive tag to indicate if it's present within the urine. So let's look at the EMIT analysis because this one is typically used, but it's specifically, again, an antigen antibody reaction um, that is used for detecting blood drugs in both urine and blood. And basically, they create these antibodies that bind to specific drugs. Um, and then they add that to the blood sample or the urine sample. And if the drug is present, it will cause clumping just like the blood typing test is. But what you need to remember is that these drug antibodies are not natural, okay? They don't occur in nature. And so by combining a drug with a protein, they can then inject that into an animal, which is typically a rabbit. And basically, it acts as an antigen. And so what it does is it acts as a foreign object in the animal, and so the animal's body starts producing specific antibodies for this now um, drug protein. Well, then what they'll do is they'll remove that um, serum from the animal and use that as the anti-serum to that drug anti-serum to test the blood or the urine of the individual. So it doesn't hurt the animal, but we're using the animal to produce a specific antibody so that we can then test for that drug in an individual. This ends lecture 9-1. There will be a quiz next class.